Hi, I'm Nathan Chrismas, and I'm a postdoctoral research associate at the Marine Biological Association of the UK. And this talk is about some of the work that we've been doing here on the diversity of photobionts in the marine lichen Lichina pygmaea. Lichina pygmaea is a saxicolous fruticose cyanolichen within the family Lichenaceae, and it's distributed widely across European North Atlantic coasts, where it's found fairly commonly on exposed rocky shores, including in the area surrounding Plymouth, where the MBA is located. It's also one of the few true marine lichens being found exclusively in the eulittoral zone, where it's fully submerged in seawater twice a day, for anything from a few minutes to several hours, depending on the state of the tide. Previous studies have indicated that the main photobiont of Lichina pygmaea is Rivularia, which is a filamentous heterocystus cyanobacterium within the Nostocales. And these studies used Sanger sequencing to directly target the main cyanobacterial photobiont. But we were interested in using a different community-based approach to see if there was any way we could determine any finer scale community structuring within the thallus. And on top of this, we were also interested in looking at the diversity of eukaryote algae associating with Lichina pygmaea, since these lichens are usually found growing alongside numerous seaweeds on the rocky shore. So we carried out a metabarcoding survey of 3 Lichina pygmaea phalli, using the 16S marker gene for bacteria and the 18S marker gene for eukaryotes, and comparing the communities from within the Lichina pygmaea thallus and communities associated with the thallus exterior. And what we found was that there were distinct communities of bacteria and eukaryotes inside and outside of the thallus. In line with our understanding of Lichina pygmaea as a cyanolichen, about half of the bacterial community in the thallus was attributed to cyanobacteria. But what came as more of a surprise was just how much eukaryote algae was present within the thallus. Over half of the intrathalline eukaryotes belonged to the Ulvophyceae, which are a common rocky shore green algae. So what exactly is going on here? To get a more detailed idea of which lineages were enriched within the thallus, we compared the differential abundance of sequences between the interior and the exterior. And the outcome of this was that we found 4 different sequence variants of Rivularia enriched in the thallus, which is expected as this is the main photobiont. But we also found 2 sequence variants related to Pluricapsa, one of which was extremely abundant. And this is very interesting, since Pluricapsa has never previously before been reported as being in association with Lichina pygmaea. And furthermore, we found 1 sequence variant each of the Ulvophyceae Pulbrogia and Bladingia enriched in the thallus. Again, no previous evidence of green algae in association with Lichina pygmaea. And we think this is really exciting. We've potentially gone from looking at 1 photobiont in Lichina pygmaea to as many as 4, 2 of which are in a completely new kingdom for this lichen. Looking at the cyanobacterial ASVs in a bit more detail, there are a couple of things to take note of. Firstly, different sequence variants are enriched in different thalli, suggesting that the cyanobacterial photobiont populations are independent in each thallus. Second, these Pluricapsa-like ASVs derived from a mixture of marine and freshwater or terrestrial lineages. And this is really interesting. It may be a sign that Lichina can recruit secondary photobionts based upon the amount of freshwater runoff at particular locations. The The story with the green algae is slightly different. The same ASVs appear repeatedly in all three phalli, and they're both marine algae, typical of the intertidal. Both had very low abundance in the exterior of the thallus, indicating that they are a genuine component of the lichen community, rather than just being taken up from the environment. And what's interesting to note here is that both of these algae form known associations with marine lichens in the Eukaryote, such as Hydropunctaria. And what we may be seeing here is evidence for photobiont sharing within the lichens of the littoral zone. So to conclude, photobiont diversity in Lichina pygmaea appears to be much greater than we previously thought, bringing in multiple cyanobacteria and green algae, with the presence of mainly marine photobionts supporting the concept that lichenized fungi rely on locally adapted photosynthetic partners, which they may share with other marine lichens. And this opens up some interesting questions to take forward. Like, how are these novel photobionts integrated into the thallus? How are they related to the photobionts of other lichenized fungi in the same environment, such as Hydropunctaria and Ballenbergiella? And of course, this all feeds into updating our understanding of the nature of the lichen symbiosis, moving away from a simple two-way partnership to a much more complex ecology of interacting organisms. I'll leave it there. Thank you very much.